Hey everybody, so this is Katie here. So this is going to be our new normal for labs going forward. Unfortunately, due to the pandemic, we're no longer meeting in person, and so our labs will have to be virtual. So you'll get to see more of me like this. And so it's going to be the same kind of format. The only thing that's going to be different is we won't be meeting face to face. And so today we're going to be talking about lab. And so what I have set up for you today are your reminders. So make sure today to turn in your lab number 7, which was our Hardy-Weinberg lab. I know we had an extra week of spring break, and there were a couple reasons for that. The primary one being they wanted to give us enough time to prep for our virtual classes to get started. And so it was a bit of a scramble, but this is the best we could do with the situation that we have. Nobody signed up for this, so including me, and so I hope that we can make the best of a bad situation. So anyway, make sure you turn in your lab number 7 Hardy-Weinberg lab. We did that a few weeks ago now. So you have two options to turn it in. You can either take a photo of the front and back of the worksheet that you guys did in lab, or you can scan it front and back, make sure your name is on it, and you can either submit it in Dropbox to me under lab number 7, or you can email it to me directly. So make sure you guys get that turned in so you don't lose easy points. The second thing to remind you guys of today is that module number 7 quiz is due today by 6 p.m. So no longer will they be taken in the testing center. Instead, they are now open book and 20-minute tests or quizzes. And so you'll have approximately 2 minutes for each quiz, but 20 minutes total basically. So make sure you're not going over your time. There is a reminder that will click up into place to remind you that your time is running short. So, and that module number 7 is all about microevolution. So if you remember all the different types of microevolution, the different types of selection that we went over in the last week that you saw me before spring break. And so just make sure you read your readings for that, and hopefully you guys will do well on it. Okay, so reminders for next week. So this is going to be Tuesday, April 7th. This is looking ahead, so just to keep everyone on task. Your module number 8 quiz is going to be due on April 7th, and also this lab that we're doing today, lab number 8, Island Biogeography, is going to be due on April 7th as well. So the lab that we're doing today will be due 1 week from today on Tuesday, so you're going to have to turn that in to me. Okay, so today, Lab 8, Island Biogeography, can be found in Cobra, and I've also attached it to your email that I sent out to all the students in this class in Bio 107. So either download it from Cobra or download it from your email where it was attached. And it's several pages long, so make sure you download it. You can save it to your computer, just save it. Or you can print it out. That'll probably be the easiest option if you wanted to print it, but it's really up to you. Okay, so today for the lab, there's 4 parts of it. Now I know it's a few pages long, so it's going to seem a little daunting, but I'm going to try to break it down for you as best I can, so hopefully there's going to be no questions. So part 1 basically is going to be looking at phylogeny based on geographic distance. Part 2 is going to be looking at phylogeny based on geological history. Part 3 is going to be looking at phylogeny based on your morphology. And part 4 is your phylogeny based on your molecular genetics. Okay, so we're going to go through each part of that just so you guys don't get confused, because I know there's a lot of new terminology here. So hopefully I'll I'll make it a little bit easier on you. So we'll talk about what phylogeny is in a second, but I just want to point out that the lab today, we're going to be basically looking at island colonization of lizard species. And there are some general principles to keep in mind when we're talking about island colonization. Okay, so the closer the island is to another landmass, typically there is a higher probability of colonization. So the closer an island is to a continent or a large landmass, the more likely you're going to see species existing on that island. The second thing is the older the island is, the more likely it will be colonized. So if the island is relatively old, you'll likely see species that are living and existing on that island as well. The third thing is the larger the island, the more species there are likely to be established. And so you have a lot more space, you have a lot more probability that there will be a lot of different species there that will be living and inhabiting that particular island. The fourth general principle here is that geographic isolation reduces gene flow between populations. So when you have some sort of geographic isolation or barrier like a mountain or the ocean, it's going to reduce that gene flow between those populations on those islands or on either side of the mountain or whatever we're talking about here. And then lastly, over time, you'll have populations that become genetically diverse from the parent population due to a variety of factors such as natural selection, mutations, and genetic drift. So time will essentially change those populations to make them different than the original parent population from which they came. Okay, so I use the term phylogeny. So what is phylogeny? So basically we can define phylogeny in two different ways. So for the sake of the lab here today, we're going to look at both. And so phylogeny is technically the evolutionary history of a certain kind of organism. So in today's lab, we're going to be looking at lizard species. It can also be defined as the evolution of a genetically related group of organisms. So the two different definitions, not too different. Basically, we're looking at the evolution of an organism, genetically related over time. So in the lab, when they ask you to draw the phylogeny of the lizard species, this is kind of what they're expecting for you to draw. So it's going to look like this. So in this example I have here, this is not in your lab, this is just something I put up here to make it a little bit easier for you guys. But so this is the phylogeny of apes, great apes. And so you can see over 50 million years ago on the far left of this slide here, you have the more primitive or prehensile tail ape species. And as you go to the right-hand side of this phylogenetic tree, you can see the phylogenetic tree gets younger, and you can see us humans over on the far right side. So we are one of the newest species here, whereas on the far left you can see the older species, or prehensile tail monkeys, New World monkeys. And on the far right you see the youngest of the species, gorillas, chimpanzees, bonobos, and humans. So keep that in mind when you're drawing your phylogenetic trees, and they'll ask you to draw your phylogenetic tree or your phylogeny for these lizards several times. And so you're going to be doing those a few times, and they might all look different each time you do them, which is kind of the point, because we're using a lot of different things, those different parts, geology, geography, morphology, and genetics, to determine the phylogenetic tree 4 different times, basically. Okay, so these are the lizards that we're going to be looking at. There's a few different types of these lizards, a few different species. So Galatea is the genus of these lizards here, and so we'll be looking at the genus and a few different species of these types of lizards. So these lizards are going to be found on the Canary Islands, so this off the coast of Africa, and you can see in this first map here, you can see all the islands are labeled, and you can see the coast of Africa on the right side of this image, and you can see our little measurement bar of 100 kilometers. And so keep that in mind, because it's going to ask you to measure the distance and to look at the different islands and see which one you think is the youngest, which one you think is the oldest, and why. And that's going to play an important role in determining what species of lizard you think is the newest, or what species of lizard, or the youngest species, and which species of lizard is the oldest type of species. So in this map here, so we're going to be looking at part 1. So those two maps, the first map I showed and this map. So part 1, we're going to be looking at phylogeny based on the geographic distance. And so you can see here that there's all of these different islands. You can see that depending on the type of lizard and how the map is colored in this specific example, now this is in your lab, so you'll be able to see this image again. Depending on the type of the chart here of the maps and of the key, you can see what species of lizard inhabits which islands, and you're going to be determining the phylogeny of these different species of lizard based on geographic distance of these islands. So all of these lizards have colonized these different islands. You're going to be looking at the distribution based on what you think is the oldest and what you think is the newest. Okay, it doesn't look like my map showed up here kind of okay. Anyway, so this is basically showing you part 2. This is going to be the phylogeny based on geological history. So it shows you in this table here the island name. It shows you how old the island is, and so you're going to be drawing a phylogeny of lizards based on how old the particular islands are and who colonizes those islands. So try to keep that in mind when you're drawing this phylogenetic tree, which lizard is living on which island. So that's going to be important for drawing your phylogenetic tree for the part 2. So make sure you are looking at which island, how old the island is, and which type of lizard of the species live on those particular islands. Okay, for part 3 here, you're going to be looking at phylogeny based on morphology. So morphology is the relative size or the physical makeup of the lizards in this case. And so you can see in this image here, or this figure, the sizes of the lizards on each island varies a little bit. So you can see some are larger than others and some are smaller. So you're going to be drawing a phylogenetic tree based on the morphology. And there's a little bit more detail also in the lab itself, but I'm not going to give it all away because I want you to kind of determine who you think is the most closely related based on their morphology or their relative size for each of the populations shown here. Okay, and the fourth part here that we're looking at, so this can get a little complicated. So I have kind of cut some pieces of the lab out and put it on my PowerPoint slide here. So now we're looking at, for the fourth part, the phylogeny based on molecular genetics. And so it's going to ask you to fill in a table, and it's going to give you all of this code of these base pairs. So if you remember the base pairs that we talked about several modules ago, we're going to be looking at those again. And so in the different the code here, we have, I believe, 7 different species in your lab. But here I've only cut the first and second species that they've listed, just to make it a little bit easier for you guys to look at. So here we have the G. stellini for under 1A, and under 2A we have G. atlantica. And so I cut the second, the third, and the fourth parts of their code out as well. So that's your 1B and your 2B, your 1C and your 2C, and your 1D and your 2D. So that code is all for that same species. So 1A, B, C, and D is all for the G. stellini species. So the line of 2A, B, C, and D is all for the G. atlantica. So when you're looking in to fill in the chart, make sure you're looking at the right line of code. So now it's going to ask you to compare the base pairs or the differences in the genetic code here. And so in order to do that, what you're basically going to be doing is counting and seeing how many differences there are in the base pairs that we have. So I've gone ahead and cut the 2 lines out. And it might be a little bit easier if you guys wanted to print out all of your papers and kind of cut it and line them all up in a row together. That might be easier. But I was able to just kind of look at it and make sure I was looking at the right lines and count them that way. And depending on the chart on the next slide, which we'll look at here, it'll tell you which 2 you are comparing. So make sure you're comparing the correct 2 species of lizard. So in the first case here, I'm comparing these 2 species, and you can see the first line of code for G. stellini is all spelled out. All the base pairs are there. The A, G, and the A and T and the C and G are all spelled out for you. Now you can see the difference in the base pairs. The ... dots indicate that those base pairs are exactly the same, but where you see a different base pair here is one of the different, for that particular species, one of the different codes for that species. So in this case, you can see the G. atlantica has a different base pair. It has a different T, which is the first T there, the second T that I have circled in red, the third T I have circled in red, the fourth one is an A, the fifth one is a T, and the sixth one is an A. So I've circled all of the different ones here in this image. Now if you go through and look, you can see that if you circle them all and count them all out, that there's actually going to be 36 different base pairs in these 2 particular species. So they vary, they differ by 36 base pairings. So that's how much of a difference there is between these 2 particular lizard species. Okay, so in the next chart here, you can see that I have my G. stellini, and oh, I circled the wrong one here. I'll fix it. So you can see in this chart here, I have the G. stellini and I have the G. atlantica. So I compared those 2 particular species, and they differ by 36 base pairs, which is what I've counted out. And so make sure that you're comparing the right 2. So this next example here is G. atlantica compared with G. galati, and that's on the Palma Island. So make sure you're looking at the correct island that they're inhabiting. Make sure you're looking at the correct island that they're living on. And then that way you can make sure you're comparing the correct 2 species. Okay, so for that G. galati and that G. atlantica, you can see there's 25 differences in those base pairs. So that's just to give you an idea. And if you go through and count all the differences between those base pairs, you can see that there is in fact 25 differences. So you need to fill in this chart for all the other species that they want you to compare. Okay, so it's pretty straightforward. One of the most important things I want you guys to keep in mind is now that we are all virtual, it's going to require a little bit more wherewithal on your behalf. So make sure that you're reading. And it might just be an extra few minutes to really read what the lab is saying, because I won't be there in person to answer questions. Now, I will be available via email, and I'll be checking and basically chained to my home office here for a vast majority of the time, since I'm trapped in my house just like all the rest of you are. So hopefully you have a lot of time on your hands and you guys are all safe, but please make sure you're reading. And then if you do have questions, just shoot me an email. I'll be around. Not going anywhere anytime soon. So, and then remember, this is due 1 week from March 31st. So it'll be due on April 7th. So just keep that in mind as well. And like I said, any questions, just send me an email, preferably not an hour before the lab is due. So just make sure you look at it before the actual due date. So hopefully you guys have a lot of time now to focus on your studies if there's nothing else for y'all to do and you're bored. So, all right. So that's all I have for the lecture for the laboratory today. Feel free to reach out, and I will check in again with you guys soon.